Good afternoon. My name is Jan Torres. I'm a veterinarian and a researcher at CECAV, Spain. And today I'll be presenting this study, which is part of my PhD. It's titled Prophylactic Novel Administration of Phages to Enhance Protection Against Salmonella Typhimurium in Broiler Chicken. The poultry meat production chain starts with a thorough genetic selection of different lines of animals, which tries to enhance productive traits in broiler chickens. After several generations of genetic selection, the resulting eggs are sent to commercial hatcheries, where they spend around 21 and a half days in an incubation cycle. Once the eggs hatch and we have the chicks, those are sent to farms, which are usually spread through wide territories. On farms, they spend around 41, 42 days, and then they're sent to slaughterhouses, where final products are processed and distributed to stores for final consumers to buy. The main concern here is that although the presence of Salmonella is a public health threat in any of these stages, its presence in hatcheries can be an even greater threat. Since the eggs from the genetic selection come from different origins and all of them meet in hatcheries, different Salmonella cerevars or strains can gather in hatcheries and then go with the animals that are distributed through territories to the farms and reach even more final products, posing an even greater food safety hazard than in any other stage. So the aim of this study was to assess the efficacy of bacteriophage application in ovo as a preventive measure to mitigate Salmonella transmission from hatcheries to rearing farms. To do so, 132 eggs were incubated as they would have been incubated in commercial hatcheries for 21 and a half days. And then throughout the whole process, the eggs were monitored to detect any non-embryonated or dead egg. Then on day 18, prior to the hatching window, which is obviously by the end of the cycle, the eggs were inoculated with a phage cocktail. The cocktail was composed of these three phages, CECAV 41, 48, and 50. They were phages that had been previously characterized. They were strictly lytic and targeted Salmonella Typhimurium. Then, in this case, the phage cocktail was administered in the amniotic liquid, as depicted there. First hole was pierced through the eggshell on the air cell, and then the phage was inoculated. In this case, we inoculated half of the remaining eggs on day 18. That half was inoculated with 10 to the 8 PFUs per egg and became the treatment group. The other half was not inoculated and became the control group. So once the cycle had finished, the hatched chicks were transferred to experimental farms. In the experimental farms, they were reared for 1 week. As I said, the cycle usually takes around 41 days, but we focused on the first week because it's when they have a less mature immune system. So it's when they are more susceptible of acquiring pathogens or bacteria that can hinder the productive process later on. So during that first week, both groups, the treatment group, which let's remember was inoculated with phage, and the control group were infected with Salmonella. They were orally infected with 10 to the 4 CFUs per animal, and it was a Salmonella Typhimurium strain. Then, throughout the rest of the week, Salmonella concentration was assessed, sampling, taking two types of sample, environmental samples, which were boot swabs, and to control Salmonella colonization within animals, cecum samples from the animals were taken. Here we have the results from the first part, which is the incubation cycle. Here, depicted in blue, we have the hatching rate expressed in percentage of the hatched eggs, and then in brown, we have the unhatched eggs. So as we can see, the non-inoculated eggs had a hatching rate of around 85%, and the phage inoculated eggs a hatching rate of around 95%. Then the statistical analysis enlightened that there were no significant differences between groups, and the overall result was around 80%, which is what we would have expected in a regular incubation cycle. Here we have the results regarding Salmonella concentration. At first sight, we see that the red bars, which represent the control group, are always above the treatment group, which are in green. when we focus on boot swabs, which were the environmental samples, we can see that there were statistical differences on day 2 and 3, and those were around 4 log units compared to the treatment group. Then when we look at the CECAV samples, we can see that the statistical differences were constant throughout the whole first week, and the major differences were appreciated on day 2, where we had a 5 log unit difference. So bearing these results in mind, the conclusions of the study were that phage administration proved to be harmless to embryonated eggs, that in novo phage application reduced Salmonella Typhimurium colonization during the first week of the rearing cycle in the animal CECAV, and that in novo phage application reduced Salmonella Typhimurium colonization during the first 3 days of the rearing cycle on the environment. All this study and further data have been recently published in an article, which also has additional in novo trials that we have performed, which you can find in the QR. And before I finish, I'd like to thank all the staff at CECAV who participated in this project, and my supervisor, Sandra Sevilla, and all of you for your attention. Thank you very much.